Hello, this is the fifth part of the series on fMRI data analysis, focusing on group analyses. For typical fMRI processing or analyses, we first process the data from each individual subject, which is sometimes referred to as first-level analysis. This consists of the pre-processing steps, discussed in my earlier lecture, and then determining the activation amplitudes or beta weights and any general linear tests and statistics. For the group analyses, sometimes referred to as a second-level analysis, we typically compute the statistics from the beta weights, that is the estimated activation amplitudes. There are also programs that use both the estimated beta weights and their statistics in the group-level analyses. Some examples or hypotheses that we might be interested in testing with group analyses include: what brain areas are active during a particular task? In this case, the null hypothesis is that the average effect is 0. What brain areas are more active in one task versus another? In this case, the null hypothesis is that the beta weights, the amplitudes of these two tasks is the same. Or we might be interested in finding out what brain areas are more active in one group versus another for a particular task. In this case, the null hypothesis is that the beta weights of these two groups are the same. First, some definitions. A group or statistical analysis can have different factors. These factors can be either categorical or quantitative. So for example, they could be either gender, group, or conditions, some examples of categorical tasks, or more continuous, such as age, IQ, stress level, reaction time, and so on. The factors can be fixed or random. Fixed, that's generally something we have control over, such as the group or the condition. Or they could be random, such as the subjects that they are coming, basically a random sample from a particular distribution. We might also be interested in certain covariates, which can be either categorical or quantitative. Categorical covariates are typically converted to some number, like 0 or 1 or -1 and 1, before including them into the analysis programs. And we might also be interested in certain interactions with two or more factors. That is the difference in one of these factors dependent on another factor? I'm going to be discussing these concepts using an fMRI paradigm that we ran several years ago. In this paradigm, participants were asked to perform a verbal fluency task. So for example, they saw either a single letter or a single category on the screen, and they were asked to name as many words as possible that started with that letter or that were part of that category. Alternatively, they could have seen 2 letters or 2 categories, and in that case, they had to switch back and forth between both of those letters or between both of those categories. In addition, we had 2 control conditions. One, where the word months appeared on the screen, where they simply had to say the months of the year out loud. This is an overlearned category, for example, January, February, March, April, May, and so on, that really somebody doesn't have to think very hard about. And finally, we also had a control condition just looking at a fixation spot. The reason for including this control condition was that we were ultimately interested in finding out what are the parts of the brain that are engaged when a person is really trying to think of a word that starts with a particular letter or that's part of a particular category, compared to when they don't really have to think about it, but controlling for a lot of the other things, such as seeing a word on the screen or the actual speech output. This particular task was presented in a block design with 10 task blocks per run, each of these blocks lasting 10 seconds and consisting of one of 5 conditions. Each of these conditions were presented twice during the run. So the first question we could ask is, what areas of the brain are active? That is, for example, during the single letter condition. This is an analysis with one group, one factor, one level, where the null hypothesis is that the amplitude of the single letter condition is 0. We can reject the null hypothesis in the colored areas that are shown here. Next, we could be interested in finding out what areas are more active during the single letter condition versus the control condition, the months. This can be done using a paired t-test. That is, we have these conditions available for every participant, and then we can compute a paired t-test between the single letter and the months condition. Alternatively, we could have computed the general linear test between these 2 conditions in each individual subject and then ran a 1-sample t-test of that general linear test across the group. Next, we could be interested in on average, what is the difference between all of these different fluency conditions, that is the category and letter regardless of switching, versus the months control condition. In this case, our null hypothesis is that the average of these category and letter fluency conditions is not significantly different from the months, that is, that the difference between the two is equal to 0. And we can reject that null hypothesis in the colored areas below, where the red areas indicate regions where the category and letter fluency, the amplitude of those conditions are greater than the amplitude of the activation during the months condition, and the blue indicating areas where the activation is greater in the months condition. Notice that this particular task shows that the left inferior frontal gyrus shows greater activation during this condition, during this task where you really have to try to think of a word. In addition, you also see a region in the fusiform gyrus showing that, whereas areas in speech production, as shown here in blue, actually show greater activation during the months condition. And that's because participants in general were generating many more words during that particular condition compared to when they were trying to think of different kinds of words from the either letters or categories. For this particular study, we actually had two groups that we were looking at, individuals with autism spectrum disorder versus typically developing controls. And we could also ask, and we were interested in knowing what areas are activated differently in those individuals with autism spectrum disorder versus neurotypical controls. And that's what we can see here with a two-sample t-test comparing the two groups, and the regions in color showing the main effect of the group, that is the individuals with autism spectrum disorder versus typically developing controls, showing differences in the left inferior frontal gyrus and in the fusiform gyrus. We can also be interested in interaction effects. This could be, for example, within each individual subject. That is, are there brain areas where the activity during the switching condition, where you have to switch between two letters or two categories versus just a single one, is different for the letter versus category conditions. This is sometimes known as a two-way repeated measures ANOVA. And you can see in this particular condition, we see slightly greater activation in a single letter versus a two letters, but slightly greater activation for two categories versus one category in this particular brain area. And finally, we can also look at interactions across the groups. So in this particular condition, we were interested in knowing whether the category or letter fluency was different in those individuals with autism spectrum disorder versus typically developing controls. And we did find a region in the left inferior frontal gyrus where we saw that difference. That is particularly a decrease in the activation amplitude in the category fluency in the individuals with autism spectrum disorder. Now, trying to interpret interactions from more than two factors can be quite difficult. So for example, in this case, we could actually see differences in the letter and category conditions with switching or not switching in individuals with or without autism spectrum disorders. Trying to interpret these can often be very difficult, and perhaps some solutions for that would be to look at pairwise comparisons and to plot the results as I've done here, and that can often aid in trying to figure out what is driving that particular interaction effect. Another very common analysis that is of great interest in functional MRI is looking at covariates. So in this particular case, we were looking at a monetary incentive delay task. This is a particular task where a participant can either win or lose money. They're first presented with a cue, whether they can win or lose money. At some point later, a target appears. They have to press the button while the target is on the screen. And if they hit the target, they either win that amount of money or avoid losing the certain amount of money. And then they get a feedback screen saying whether they won or lost or avoided winning, avoided losing, or did not win the money. This particular task was designed to be able to separate the anticipation of the potential loss and win from the actual feedback of whether they actually won or lost the money. What we were interested in is the brain activation during the anticipation period of this potential loss correlated with some other measure, in this case, early life stress. So on an individual subject basis, we calculated the activation amplitudes during these particular periods. And then in the group analysis stage, we looked for whether these activation amplitudes were significantly correlated with this measure of early life stress. That is, we added this early life stress measure as a covariate to the group analysis. And in this case, we did find that there was a region in the putamen where the correlation with early life stress where the activation amplitude was negatively correlated with this early life stress measure, such that those individuals with low early life stress exposure had robust activation in this region, but that decreased individuals with higher early life stress scores. So in summary, over these series of lectures on fMRI data analysis, I've shown you examples of the pre-processing steps that are involved, some examples of the single subject general linear model, where the steps are really to choose the model, fit the model, get the beta weight, and get some statistics from this, and then some examples from the group analyses.